upon reflection, I thought that would really be helpful for you to see, because then you'll understand how these covalent bonds formed between amino acids within a protein structure lock that protein structure into a more rigid conformation. And when I use the word conformation, that's only to reflect the meaning shape. The proteins adopt shapes. Those shapes are called conformations. So I do want to go and show the movie. And how would I do that? Usually if I make a text box, it gives me access. Now this is a really long movie. I'm going to just kind of jump to the important parts. So here's a depiction of a polypeptide. It's really just a long string. And that's kind of the way it appears as it comes out of the ribosome. It's a long linear array of amino acids linked together by these peptide bonds. It's adopted some structures indicated by the blue and the red, but we haven't thought about those structures yet, so ignore them for now. Let's just think of it as a string. Those yellow balls are cysteine residues. They occur at random places throughout the protein, right? They don't have to be side by side to be able to interact with one another, and that's what this movie is designed to show you. So these polypeptides, they start changing shapes and forming structures, and so now you can see that two cysteine residues that weren't side by side, in fact there's a cysteine residue between them, have now formed a disulfide bond. So, oops. Here it is. So I might not be able to draw, I can't draw in the movie. Sorry. Okay, so there's the best view of that. All the balls are cysteine residues, but two of them that are separated by a fair number of amino acids, it looks to me like it's probably 50 or so, have adopted now a shape where you can see it looks kind of like a paperclip at the top, where those cysteine residues have now come within proximity of one another, very close to one another, and they formed a disulfide bond. That's a covalent bond. That's not going to change, ever. Okay. So now that polypeptide is locked in that bent configuration. It can't get out of it anymore. It'll continue to bend and twist and try to form other interactions, and now you see that it formed another second disulfide bond, which we just skipped right by. There it is. So now a second pair of cysteines have formed a disulfide bond as well, and now we further locked this polypeptide into a shape. It's beginning to take a shape. Those disulfide bonds never go away. Hydrogen bonds are easy to break, remember? Van der Waals interactions are weak, but covalent bonds are the strongest of bonds, and it would require an enzyme to separate them. So even when you boil proteins, the disulfide bonds don't break, even though all other interactions would be disrupted. The disulfide bonds are not. And we'll actually see that in the lab, because we're going to treat our lysates that we generated yesterday with a reducing agent that will break these bonds. Okay? And we have to for what we're going to try to do. So now we've adopted a second structure. So this is why these covalent bonds are so important in stabilizing protein folding. Not all proteins form disulfide bonds. Organisms that live in extreme environments, like those animals that live in heat vents, for example, they use a lot of disulfide bonds. because they need proteins that are stable even in the presence of high temperature. So they form a lot of disulfide bonds in their proteins to try to help stabilize them and survive the harsh conditions that they exist in. Okay, so that's all I really wanted to say about disulfide bonds. I just want to make sure you understand examples of how they form. So we've already talked about hydrogen bonds. I went through very carefully and defined what a hydrogen bond is. And when you look at the homework, not the homework, but the exam study guide that will be posted tonight, you'll see that that's going to be one of the questions. Define hydrogen bonds, define electronegativity. These are the important concepts that we need to understand going through this section of the course. And so it's best for you to think about that a lot so that you feel very comfortable with the idea of what those topics mean. All right, so covalent bonds, hydrogen bonds, van der Waals interactions. I'm sure you guys had this in chemistry, so I don't want to belabor it too much, but it's a really cool interaction because it can occur between any two atoms. Any two atoms can interact through van der Waals forces. That's not true of covalent bonds. That's not, and by covalent bonds, the only one I refer to is the disulfide bond. That's not true for hydrogen bonds. It's not true for ionic bonds. It's only true for van der Waals. And so this is a diagram meant to explain what these van der Waals forces are, but they're pretty easy to understand. What it's trying to show you is here we have two atoms. In fact, in this case, they're the same atom. They're two of the same atom. And they have 4 protons and they have 4 electrons. So if you imagine such an atom, if you have one in each hand and you're holding them, so my fists are now the nucleus. The protons are located. The electrons are orbiting really, really close to that nucleus, right? And so if I hold those two atoms at quite a distance apart, they cannot sense that each other's in the area. And the only way they'd sense that is by feeling their electrostatic activity. So really far apart, the atoms can't sense each other. But as you move them closer and closer and closer and closer, there becomes a point at which they can sense each other. This one's got protons in the center and electrons, negatively charged electrons, circling around that nucleus, and then so does the other one. As you bring them closer and closer, now those electrons can feel each other. There's an opposite, repulsion between them, because they are similarly charged. Hold two magnets, so the same poles are facing one another, and you'll feel them trying to push each other apart. And that's what would be happening with these two atoms. So at a fairly close distance, those two atoms actually repel one another. But imagine what happens as you continue to move them closer together. And that's what this picture is meant to show down here. So as you move those atoms even closer still, the electrons of one atom will begin to push the electrons of the other atom to the opposite side of its positive charge. So they're repelling one another, so that the electrons begin to spend most of their time away from the other atom because of this repulsion. And what happens when those electrons are repelled is now the electrons from one atom are closer to the positive charge on the other atom. And that actually becomes attractive. So now we've got opposite charges getting close to one another. And so that becomes an attractive force. And that attractive force only happens when you're really close to one another. In this case, it's designated as 5 nanometer distance. That's called the van der Waals radius. Okay, if you kept moving the atoms closer still, now the two nuclei will be getting close enough to one another that they'll start repelling one another. So there's a balance. There's a wheelhouse. Everybody play baseball. So the pitch comes right down the center. It's easy to hit. It's right in their wheelhouse, right at that very defined radius called the van der Waals radius, that's where the attraction is most strong. You shorten that distance, make that radius, those two atoms closer together, and now they start repelling one another. Take them further apart, and now the electrons will come back out and they'll repel one another again. So really far apart, repelling. Now they become attractive, but then you bring it even closer still and they'll start repelling one another. You can see why it doesn't matter what the identity of the atoms are, because all atoms have positive charged protons and they have negatively charged electrons around them. So this idea of attraction and repulsion and van der Waals radius is true for all atoms. The radius will change depending on the size of the atoms that are involved. This is actually a big driving force for protein folding, as you probably will imagine, and I'll bring it up again when we look at the structure of a protein. I think I just changed the position of that slide. Okay. All right, so we did covalent bonds, we talked about hydrogen bonds, we talked about van der Waals interaction. We're going to think about hydrophobic interactions. I've already described this to you the last time we were here. Here's the picture that goes around along with it. So in this case, we've got two hydrophobic molecules indicated by these brown lozenges. And they're sitting in water, and then the blue dots are meant to represent water. These hydrophobic molecules can't interact with water. And so water has to form this really kind of structured network around it. And probably these are held together by hydrogen bonds with other water molecules, but they're not interacting with the hydrophobic molecule at all, which means these waters then become very structured around that molecule. And same thing with this one. And in our simplistic view of how this happens, we can actually look at the number of water molecules that are structured around that hydrophobic molecule that can't interact with it. And in this case, I see 3, 6, 8 for each molecule. So there's 8 waters that don't have high degree of freedom in the presence of each one of those molecules. So it's 8 times 2, 16 molecules that are restricted in what they can do in solution. If instead the two hydrophobic molecules can interact with one another, which we know they can do because they're made of atoms and therefore can at the very least interact using van der Waals attractions. Now the number of water molecules that are structured around those molecules is reduced. In fact, all of the water molecules that were surrounding the sides of the hydrophobic molecule that is no longer exposed to water have been freed up. And so if we count, we should get a smaller number. So there's 2, 4, 6, 8, 10 water molecules here, whereas there were 16 over here. So that difference in 6 water molecules that can now feel free to move around in solution and interact with other water molecules actually reduces the free energy of the system. Okay, yeah.
Why again do the water molecules go away?
Because there's no room for them anymore, right? These two molecules, the faces of these two molecules, which were over here, let's see if I can grab a different color. The two faces that were here and here, exposed to water and had structured water molecules around them, these 6, presumably, those 6 water molecules no longer are forced around the hydrophobic molecules because the hydrophobic molecules are interacting with each other.
Okay.
This is called the hydrophobic effect. It's not truly a force at all, right? It's just a reduction in free energy, which all organisms are trying to achieve. Okay, here's another picture of it. If you wanted to look at side groups of polypeptides and ask how does this really relate to a protein structure, here's a methyl group. We know methyl is the side group for alanine. So here's the side groups of alanine with all the water molecules structured around them. You can see how the water molecules are forming hydrogen bonds with each other, but can't interact with the methyl group, and so there's potential hydrogen bonds that aren't being used. Here's one, there's two more, there's two more, there's, so there's all these potential hydrogen bonds that can't form for both of them. But if you stick these together where they can interact through van der Waals interactions, now there's less structured water molecules around. Okay? All right, so let's see. So now we've talked about covalent bonds, hydrogen bonds, van der Waals, hydrophobic effect. That only leaves one more. Which one is it? I don't know. I guess I don't have a picture. Let's see. We can draw a picture of it. If we think of the side group, one of the side groups we're supposed to memorize. Is there a charged amino acid we're supposed to memorize the structure for? No? I know there's one. CH2. I think we chose this one. We chose this one, lysine, and I believe that's the structure of lysine, but don't hold me to it. And then we probably were supposed to memorize a negatively charged amino acid. Aspartic acid, yeah. Oh, good. So let's see, that one's got a carboxyl group at the end that's negatively charged, and then a bunch of CHs, I don't remember how many. I'm just going to say it's at least that many. What did we say, aspartate?
Mm-hmm.
Is that what you did? Aspartate, that's an E, I think, right? So now we've got two amino acid side chains that have full charges on them. And that we all recognize as being an ionic interaction. There's the ionic bond we'll use as our example. This is the one we've talked about a lot before, and I've used this analogy of sodium and chlorine being side by side and forming these salt bridges, and that's where these terms salt bridges came from table salt. That's a really strong interaction in a dry environment, and so is this. This is a really strong interaction in a dry environment, like the inside of our protein. And I touched on that already. I'm not going to re-lecture that. All right, so I think this is an animated slide that's worth animating, so we'll take a look at it. It's going to highlight the different types of bonds that can form in a polypeptide. The purple string in this case is the backbone of a polypeptide. When we talk about the backbone of a polypeptide, we're talking about everything except the side groups. Okay, and that's the backbone. They're all covalently linked one to another in a long string. That's what we'll call the backbone. So the purple string represents the polypeptide backbone. And then we can look at examples of interactions that would occur within that polypeptide, whether it be between the backbone parts or between side group parts or some sort of mix together. So we should recognize this one. This is a hydrogen bond. It's an electrostatic oxygen. Should be a second bond there. I don't know why there's not. And a hydroxyl group, and that hydroxyl group looks like it belongs to serine. We can also see hydrophobic interactions between these two side groups, and those two side groups we should recognize as valine, because that's one of the structures we're supposed to remember. Remember, it's the shape of a V. Here's a disulfide bridge between two cysteines. And here's an ionic bond between two side groups. Probably lysine, maybe aspartate. I don't have those structures memorized. That's not my kind of Okay? Here's another way of looking at those. There's a lysine and a glutamic acid forming a salt bridge. Those electrostatic interactions, remember if they're inside the protein, they become really, really strong and stabilizing. We see van der Waals interactions and hydrophobic effects working in the middle here. And we can see hydrogen bonds over here. All of these are stabilizing the structure of that protein. Yeah.
Is there confirmation that a protein ultimately adapts the one with the lowest free energy?
I'll have a slide for that. And the answer is, proteins breathe. So they'll lock into a structure and then never move again. But they will oscillate right around that lowest free energy. Okay? Hydrogen bonds are broken and formed all the time, just like in water. It's the same is true in proteins. So proteins can kind of vibrate almost, because they're living structures. But they will be folded down to their lowest, essentially lowest energy conformation. And I'll show you a good graphic for that soon, maybe not today. But I want to point out, while we got this slide going, and I think I got to get out of PowerPoint mode and show. The thing I do want to point out is this one. This is the hydrogen bond. We all recognize it, but what's important about this particular one is that we recognize, at least I want you to be able to recognize, that that hydrogen is part of the polypeptide backbone. And the interaction, the hydrogen bond, is with a carbonyl group that's also part of the backbone. And if you remember, the structure of a polypeptide, which I'll draw as just a generic polypeptide with side groups of R, those groups occur regularly at regularly spaced intervals. I just draw that to that hydrogen. Right. So here's a couple of them. You should be able to draw polypeptides pretty easily, because they're just a repeating structure. But here's a carbonyl group. We've looked at these before. Here's another one. There would be another one here if we had another amino acid. Here's an amine group, and here's another one here. So the hydrogen bonds between these two portions of a polypeptide are occurring from the amine and the carbonyl groups that are present on the backbone. And those happen every single time you put an amino acid in. Those functional groups appear on every single amino acid. Okay, so there's lots of opportunities for hydrogen bonds within the backbone itself. These interactions are not restricted to side groups. You should understand that. Appreciate that. Okay. So there's a picture, Tom, Dr. King, I know talked to you about before. If we think about the types of side groups that are on the amino acids and where they're likely to end up in a folded protein, we know that the polar groups and the charged groups are more likely to be on the outside of the protein where they can interact with water. And we know that the nonpolar groups are more likely to be found inside the polypeptide. And that's all this is going to show you. Here's an unfolded polypeptide here. And as it adopts its structure, which we'll see in more detail soon, all the polar side groups, or most of them, which are now blue and yellow, appear predominantly on the outside of the polypeptide. Whereas the nonpolar ones, which are green in this example, are predominantly found on the inside of the protein. Does that mean they're all found on the inside of the protein? The answer is no. They're predominantly found on the inside of the protein. The charged and polar ones are predominantly found on the outside of the polypeptide. But again, proteins are going to fold to achieve their lowest energy state, and that might require that some nonpolar groups be facing out. There's only so many different shape changes you can explore to achieve this lowest energy state. And we'll actually look at that too. We'll watch a protein as it folds. This is another cartoon that we'll look at. I'm going to figure out how to start these things. This protein actually has a name. It's called granulocyte colony stimulating factor, but it doesn't really matter. What they're trying to show you is that it's a linear polypeptide when it's first made, and there's the identity of all the amino acids. You can represent them any way you want, letters, balls, strings, doesn't really matter. that protein will start to try to adopt secondary structures as it enters water outside the ribosome. It's already formed a bunch of them now. These are going to be alpha helices. But then it folds on itself even more still to form this low energy state. The amino acid on each end don't necessarily end up really far away from each other, and that's what this cartoon is meant to show you, is that the folding occurs so that amino acids from any part of the protein might end up being next to one another. So let's look at some of those highlights. So now the purple, the blue, the purple is at one end of the polypeptide. Let's see what color the other end is. It's red. So now the purple and the red, which were at completely opposite ends of the unfolded polypeptide, are now, as a result of folding now, finding themselves in close proximity to one another. And you can see it here pretty well. The purple end and the red end are now actually side by side as a result of this folding process. So that's one concept you should appreciate, is that even though they're at the ends of a linear polypeptide, they don't necessarily end up that way. It all depends on the folding characteristic of the protein. But then the other thing that I want you to appreciate is this transition. Let's see. I'll start the movie again and stop it when I like it. If you watch this thing spin around, it looks like there's a lot of empty space, particularly on the inside of the protein. But then they start filling in the orbitals of all of the electrons of the amino acids that are part of that polypeptide. And you can see these polypeptides are not easily accessible. The inside of that protein is a dry environment. There's an outside that has lots of ridges and shapes, and that's going to be important for us to think about, because we know proteins can bind to other things, including sugars, including other polypeptides, including DNA, and they need to have surfaces through which they can have those interactions. So proteins have these unique shapes and characteristics that are going to be important for their function on the outside. But the inside of the protein is usually just there to stabilize the outside. And it's a hydrophobic environment in there. Okay, so you can look at all sides of that protein, and there's no way into it, right? But there are these, I always call them nooks and crannies. After Thomas's English muffin, you ever have an English muffin, you tear it apart, and there's always, they hold the butter really good. And that's what these things look like. There's all sorts of depressions and peaks and valleys. That shape allows the protein then to have a function. And the function might be to bind DNA, and if it was, those nooks and crannies would be able to interact nice and tightly with the DNA molecule, where its nooks would fit with the DNA's crannies, and vice versa. Does that make sense? They fit so perfectly that they build up this strength of an interaction that allows them to remain bound to one another. And those are going to be hydrogen bonds, not going to be covalent interactions, but they'll be all the other four interactions that we thought about. Okay? So those shapes are important. All right, good. So that's it.
You have to select the one. Oh.
Yeah, I think somehow I did it wrong. I don't know how I did it. Does anybody see? Somebody came and painted the board for us, so we don't see that big silver thing anymore. Okay, so here's my review slide. I'll walk through it kind of carefully so that you understand what I consider to be the important things. But remember, the study guide is published every night for you. I hope you're looking at it. And it grows every single day, and I'm putting lab stuff on it. So the questions from the quiz yesterday are on there, so you can remember what those are. All right, review. As a polypeptide comes out of the ribosome, it must interact with water. That's our guiding principle. Does it interact with water well or not? Well, parts of it we know will, and parts of it we know won't. And so that's the whole motivation for this polypeptide to fold. Hydrophobic groups won't like the water, hydrophilic groups will. We've thought about what those side groups would be. Proteins do not typically function as cells, in cells as linear polypeptides. They adopt a complex structure called a folded conformation or a folded shape, based on interactions with itself and the water environment. So it's a complex kind of dance that they do so that they can figure out exactly what shape they're going to form. Forces that drive the folding and hold the polypeptide in a folded conformation include the 5 that we've talked about. And hydrogen bonds can form between carbonyl oxygens and amine groups found along the length of the polypeptide, but they can form lots of other places. It's just important that you understand that those opportunities are found at every single amino acid. And so that's going to actually be what drives secondary structure, as we'll find out in a couple of minutes. Okay, are there any questions about anything we've covered up to this point? You don't know? Or no?
I don't know.
Huh?
I don't know.
You don't know. Okay. Good. A few more definitions for you, but they're easy. If you think about folding, protein folding. 1, 2, 3, 4. 1. Linear polypeptide, string of beads, string of amino acids on a string. That's the first structure. It's called the primary structure because it's the first structure. Primary structure is the order of amino acids in a polypeptide, from the amino terminus to the carboxy terminus. Yes? Yes, good. Secondary structure, the second structure that can form as the polypeptide comes out of the ribosome are things that we're going to talk about next. They include only 2 structures. The alpha helix here, alpha helix, and the beta sheet. Those are the 2 secondary structures. We're going to look at those in painful detail in a minute. But that's the secondary structure. So this is number 1, this is number 2. Tertiary means 3. And that's how, after the polypeptide has come out of the ribosome and has had an opportunity to form alpha helices and beta sheets, those structures can now find each other and fold on top of one another.